IB Bio DNA Structure, DNA Replication Part 3 will focus on the details of replication. There are 5 important enzymes that play a role in the replication process. Remember that replication is when DNA is being synthesized from a DNA template. Transcription is RNA synthesis from a DNA template. Translation is the synthesis of a polypeptide from an RNA template. Keep the terms replication, transcription, and translation distinct. Watson and Crick, almost immediately upon the completion of their DNA model, proposed this theory of replication. This image displays semi-conservative replication, but Watson and Crick indicated that the direction of replication of each new strand proceeded in the same direction. In this respect, they were wrong. As DNA unwinds and each parental strand serves as a template for the building of a new strand, replication occurs in opposite directions. Keep this in mind. The 2 DNA strands of a single molecule run anti-parallel. This can be seen here. The 3-carbon of the deoxyribose sugar is here, thus this is the 3' end, and the 5-carbon of this deoxyribose sugar is here, this is the 5' end, while on the other strand, this is the 3' end, and this is the 5' end. Covalent links between nucleotides lie between the 3-carbon sugar and the phosphate group. This is your covalent bond between nucleotides, this one, as is this one, and this one. In the last slide of DNA Structure and Replication Part 2, I had this image where I introduced DNA polymerase as the enzyme that synthesized new DNA in a 5-to-3 direction, as can be seen in this very simple image. The new strand is being built from the 5' end toward the 3' end. As a note, prokaryotic replication has been studied in more detail than eukaryotic replication, thus we know more about the enzymes. DNA polymerase III is the enzyme that synthesizes new DNA in prokaryotic cells. We will get to know DNA polymerase I later in this movie. In general terms, when referring to the synthesis of new DNA, we simply refer to DNA polymerase. Here is the looming IB syllabus statement, and it's a handful. Explain the process of DNA replication in prokaryotic cells, including the role of enzymes, Okazaki fragments, deoxyribonucleoside triphosphates, and single-strand binding proteins. Use the terms replication forks, continuous and discontinuous synthesis. The enzymes of importance are DNA gyrase, helicase, DNA polymerase III, DNA polymerase I, DNA primase, and DNA ligase. DNA polymerase III, the primary enzyme in DNA replication, has 2 constraints, one of which you already know, and that is that the enzyme only synthesizes in a 5-to-3 direction. 5-to-3 direction only. The second constraint is that DNA polymerase III must have an existing molecule on which to add a new nucleotide. Okay, so what does that mean? When the 2 parental strands unwind, how does DNA polymerase III start its work? And the answer is that there is a primer molecule that must be built first by a different enzyme known as primase. And it turns out that the primer is a short strand of RNA. The other interesting characteristic of DNA polymerase III is that it proofreads the base pairing and corrects most mismatched pairs. Most. Mistakes are made during replication, and these mistakes are known as mutations. Here is a nucleoside triphosphate. This molecule is adenine. There are 3 others: guanine, nucleoside triphosphate, cytosine, nucleoside triphosphate, and thymine, nucleoside triphosphate. This molecule is the precursor to the nucleotide that becomes the monomer in the polymerization of new DNA. 2 phosphates are cleaved off to form the nucleotide, and the released energy is used to form the bonds between nucleotides. As the nucleoside triphosphate arrives to the active site of DNA polymerase, 2 phosphates are cleaved off to form the nucleotide. The energy in the bonds between the phosphates provides the energy to build the polymer one nucleotide at a time to form the covalent bonds between nucleotides. Here's an image of the polymerization that's important for you to study closely. Here is the template strand. Here is the RNA primer that must be in place for DNA polymerase III to initiate the synthesis of new DNA. Remember, the enzyme primase puts the primer in place. We can see the nucleoside triphosphate arriving, and following base pairing rules, it's a cytosine nucleoside triphosphate pairing with a guanine nucleotide on the template strand. Notice the 5-3 direction in the synthesis of the new strand. Here we can see the bond about to form between the primer and the newly arrived nucleoside triphosphate. Here we can see two phosphates being cleaved off, providing the energy for the bond in the building of the polymer, an anabolic process that requires energy. Again, notice the 5-3 direction of DNA synthesis by DNA polymerase III. Here's the simple image I used to introduce you to DNA polymerase and the direction of DNA synthesis. It's time we looked at the process with a bit more detail. In this image, we can see the unwinding of DNA. An enzyme named helicase does this work. Gyrase prevents or reduces the excessive coiling of DNA as it unwinds. You have now been introduced to 4 of the 6 enzymes, including DNA polymerase, primase, now helicase, and gyrase. In this image, we do not see the RNA primer formed by primase, but what we do see is DNA polymerase III moving along the template strand, building new DNA strands using base pairing rules, but moving in opposite directions. The synthesis occurs in opposite directions. Okay, simple enough along this strand, moving along as the DNA unwinds. But how does replication work on this strand if DNA polymerase moves in this direction, but the unwinding by helicase is occurring in this direction? Stay tuned. So here we can see the DNA unwinding. Helicase, not shown here, does this work, and the region of unwinding is called a replication fork. You can see DNA synthesis occurring along this strand in a continuous 5-3 direction. A continuous 5-3 direction. But along this template strand, DNA polymerase is moving in a 5-3 direction, but away from the replication fork. The synthesis of new DNA is discontinuous, forming fragments of DNA. This image should help you see the continuous and discontinuous synthesis of DNA along each of the 2 template strands. As the double helix unwinds, DNA polymerase synthesizes new DNA in a 5-3 direction continuously here. This is the leading strand. But on the lagging strand, sufficient unwinding has to occur to expose enough of the template to allow an RNA primer to be built and to allow DNA polymerase to synthesize a new fragment in a 5-3 direction. And fragments are built in a discontinuous manner. In this image, you can see the unwinding of DNA left to right as the replication fork formed by helicase pushes in this direction. You can see the continuous synthesis of DNA along the leading strand and the discontinuous synthesis of new DNA along the lagging strand. If the replication fork opens in this direction, along this DNA template, then the unwinding of DNA must expose a few hundred nucleotides along the template strand before replication can begin with an RNA primer forming another fragment. Here is a clean image showing the leading and lagging strands around a replication fork. The fragments are called Okazaki fragments after the scientist that discovered this characteristic of replication. In this image, helicase, not shown, is unwinding the DNA right to left over time. Here's another image of the leading and lagging strands, the Okazaki fragments as the key feature of the lagging strand. All new DNA synthesis is occurring in a 5-3 5 to 3 direction, the unwinding of DNA by helicase is occurring left to right over time. So let's look at this IB syllabus statement one more time, explain the process of DNA replication in prokaryotic cells, including the role of enzymes, Okazaki fragments, deoxyribonucleoside triphosphates, and single-strand binding proteins. Use the terms replication forks, continuous and discontinuous synthesis. Again, here are the characteristics of DNA polymerase III. The fact that it can only add nucleotides to an existing nucleotide requires a primer. It's an RNA primer, and it's synthesized by the enzyme primase. In this image, much like those I showed you just a few slides back, you can see the short RNA primers in place at the start of each strand of new DNA. Primase is the enzyme that builds the primers, but we will see another enzyme that must remove the primers and replace the RNA primers with DNA. As well, we need an enzyme to form the covalent bonds between Okazaki fragments to seal the gaps between Okazaki fragments. DNA ligase seals the gap between the Okazaki fragments. Here is a nice image showing helicase breaking the hydrogen bonds to unzip the DNA. It shows DNA polymerase III and primase. We can see the primer, the continuous leading strand, and the Okazaki fragments of the discontinuous lagging strand. In this image, you can see the work of the fifth enzyme I've mentioned as a part of the replication process. Ligase, seen here, is building the covalent bond between Okazaki fragments, sealing the gap between two nucleotides on different Okazaki fragments. Other than ligase, the only other enzyme this image displays is DNA polymerase III. Hopefully the rest of the detail in this diagram is familiar to you. Here's the replication fork, here's the leading strand, here's the lagging strand, here are the Okazaki fragments, 5 to 3 direction, 5 to 3 direction. New DNA in red being synthesized using the template strands in blue. The newly synthesized DNA is synthesized 5 to 3, 5 to 3. In this image, we can see the helicase breaking hydrogen bonds, thus unwinding the DNA. We can see gyrase relieving any supercoiling of the DNA as it unwinds. We can see primase building short RNA primers to which the DNA polymerase will attach nucleotides as it synthesizes new DNA. DNA polymerase can be seen here and here. We can see ligase building a covalent bond between Okazaki fragments. The single-strand binding proteins hold the strands of DNA open, allowing the enzymes access to the DNA. Now, the only remaining enzyme to introduce to you is DNA polymerase I, which removes the RNA primers and replaces those RNA primers with DNA. In this somewhat busy diagram, 5 enzymes are present. Gyrase is not shown. It is a good diagram to study for a clear understanding of each enzyme. I've labeled all 5 enzymes in red with a slightly enlarged font. Helicase is here unwinding the DNA, breaking the hydrogen bonds as the replication fork opens in this direction. Primase is here constructing the short RNA primer to which DNA polymerase can add nucleotides. Here is DNA polymerase III synthesizing new DNA strands in a 5 to 3 direction along the leading strand and in a 5 to 3 direction along the lagging strand. Here is ligase joining the Okazaki fragments formed on the lagging strand. And here is DNA polymerase I removing the RNA primer, as can be seen, and replacing the RNA primer with DNA nucleotides. Now, before leaving this slide, I want to note that DNA replication does not start at one end of a chromosome or the other to form a fork, a single fork. It opens as a bubble in the middle, and forks operate in both directions. In fact, in order to replicate DNA quickly, many replication bubbles form along the entire length of the chromosome. Here is another image that shows all the enzymes with the exception of ligase. Gyrase relieves the supercoiling of DNA as it unwinds. Helicase is unwinding the DNA, breaking the hydrogen bonds as the replication fork opens in this direction. The single-strand binding proteins hold the DNA open, giving the enzymes access to the DNA. Primase, seen here, is constructing the short RNA primer to which DNA polymerase can add nucleotides. Here is the DNA polymerase 3 synthesizing new DNA strands in a 5 to 3 direction on the leading strand, and over here in a 5 to 3 direction on the lagging strand. And DNA polymerase 1 is removing the RNA primer, as can be seen, and replacing the RNA primer with DNA nucleotides. Ligase, if it were present in this image, would join the Okazaki fragments here. Once again, here is the relevant IB syllabus statement that has been the primary focus of this movie. Explain the process of DNA replication in prokaryotic cells, including the role of enzymes, Okazaki fragments, deoxyribonucleoside triphosphates, single-strand binding proteins. Use the terms replication forks, continuous, and discontinuous synthesis. You need to know the roles of all 6 of these enzymes. In case you need to review the roles of the 6 enzymes involved in replication, here you go. The first 3 on this slide and the next 3 on the following slide. You can stop the movie to review or follow along. Helicase unzips or unwinds the DNA by breaking the hydrogen bonds between bases. DNA gyrase relieves the strain while double-stranded DNA is being unwound by helicase. RNA primase builds an RNA primer that's complementary to the template strand, the DNA template strand. DNA polymerase 3 synthesizes new DNA polymer in a 5 to 3 direction. Ligase forms a covalent bond between nucleotide gaps in Okazaki fragments, and DNA polymerase 1 removes the RNA primer and synthesizes DNA in place of the primer. Here is another diagram showing the enzymes involved in DNA replication. See if you can find them all, or is one missing? I'll leave the study and review of this process to you at this point. As I mentioned earlier, replication forks do not simply open at one end of the chromosome and work toward the other end. Instead, DNA is opened at a replication bubble. In fact, numerous bubbles, like this one, are formed along the entire length of the chromosome. In this image, I've once again stated the roles of some of the enzymes, and I've shown the RNA primers in blue, and the newly synthesized DNA is in red. This replication fork opens in this direction, so the leading strand is being constructed in a 5 to 3 direction, while the lagging strand for this fork is also being built in a 5-3 direction. This fork opens in this direction, so the leading strand is being constructed in a 5 to 3 direction here, while the lagging strand for this fork is also being built in a 5-3 direction. Now, don't be confused by the Okazaki fragments on each side of the templates, here and here. Orient yourself always thinking about 5 to 3 synthesis, and you will see why this pattern of replication seen at a bubble is accurate. And in this image, you can see 3 replication bubbles, 3 origins of replication along the entire length of the chromosome. As replication occurs, the bubbles expand until the entire chromosome has been replicated into 2 daughter DNA molecules held together briefly by the centromere. Remember that replication is semi-conservative, and you can see that well in this image, where the 2 daughter molecules formed after replication are each composed of a template strand, here in blue and here in red, with the daughter strand as orange and light blue here. In this image, you can see how with each origin of replication, there are 2 forks with helicase unwinding the DNA in opposite directions. At each fork, there is a leading strand and a lagging strand with Okazaki fragments. As we move into the final phases of this movie, here are 2 IB syllabus statements. State 2 differences between prokaryotic and eukaryotic DNA replication. State that prokaryotes have 1 chromosome consisting of a circular DNA molecule, while eukaryotic chromosomes are linear DNA molecules associated with histone protein. In prokaryotic replication, there is only one replication bubble with two forks opening in opposite directions. The rate of prokaryotic replication is faster because there is no histone protein interfering with the replication process. Eukaryotic chromosomes are linear molecules. Many replication bubbles are formed during replication. This image should make clear how many origins of replication do occur when DNA replicates. The numerous bubbles ultimately merge with the end result of two genetically identical sister chromatids held together by a centromere and separated during cell division. Here is a micrograph that captures a replication bubble during the S phase of interphase. The last topic of this movie revolves around this IB syllabus statement. Describe the method by which DNA is rapidly amplified for use in forensics. Refer to the use of Taq polymerase as an enzyme able to work at high temperatures. The method is known as PCR, polymerase chain reaction. The process of amplifying the small quantity of DNA that's been found at a crime scene proceeds as follows. The DNA is placed into a tube along with an enzyme known as Taq polymerase. This enzyme operates at higher temperatures. It's an enzyme that has been removed from bacteria that live in hot springs. In addition to the DNA and the Taq polymerase, there need to be nucleotides and a primer that's been constructed complementary to the gene of interest on the DNA. The temperature of the tube is increased to cause the DNA to separate. The primer binds to the gene of interest, and the Taq polymerase begins amplifying the gene of interest using the available nucleotides. So once again, PCR requires source DNA, primers, nucleotides, and DNA polymerase. We take the DNA, the small quantity collected from a crime scene, we heat the system to separate the DNA strands, we cool the system so the primers can bind to the gene of interest, and then DNA polymerase synthesizes DNA along each of the template strands. Cycle after cycle, and the quantity of DNA increases exponentially. Cycle after cycle, we heat to separate the DNA, we cool to allow the primers to bind, and then DNA polymerase synthesizes new DNA. Repeat, repeat, repeat. The quantity of DNA increases exponentially. Forensics investigations, paternity cases, and immigration tests use non-coding repetitive regions to distinguish individuals. Repetitive regions vary widely in humans. No two people, except identical twins, will have exactly the same set of repetitive regions, and hence no two people will have the same DNA fingerprint. And it is these repetitive regions that become the DNA of interest when DNA is amplified in PCR. You can see the crime scene DNA here with the repeats shown, as well as the DNA of the principal suspect and the DNA of the victim. The analysis of the DNA fingerprint from the crime scene and the DNA fingerprint of the suspect displays a match. The repetitive sequences, known as tandem repeats, are used in DNA profiling to link suspects to the crime scene. But typically, the amount of DNA sampled from the crime scene is not substantial. Thus, polymerase chain reaction, PCR, is critical to the analysis of DNA in forensics. PCR, polymerase chain reaction, provides enough DNA for analysis in terms of fingerprinting for forensics, paternity cases, immigration cases, disease, or in terms of determining evolutionary relationships, or for sequencing, or for mapping chromosomes so that we can locate the gene of interest in a genome. And that brings us to the end of IB Bio DNA Structure, DNA Replication, Part 3.